about your mushrooms quite here in our lectures.
So should we start, madam?
So we can start, Dr. Kutu.
Okay, good morning, everyone. Respected Dr. Padmini Shwai, Director, National Rice Research Institute. Today's speaker, Professor Yinong Yang from Pennsylvania State University, USA. All head of the divisions, scientists, and personnel from NRRI and other ICR institutes. Ladies and gentlemen, a very good morning and warm welcome to all of you, and a very good evening to Professor Yinong Yang. So I must say here that it is around 11:30 p.m. In USA now, from where Yinong Yang belong. So I must thank him for being with us and staying up so late for this lecture. You know, we assembled here to celebrate something very special to us, India's independence. Azadi Ka Amrit Mahotsav. Literally, it means the grand celebrations of India's independence. The Mahotsav is a series of events being organized by the government to commemorate the 75th anniversary of India's independence. It will be celebrated as a people's festival in the spirit of people's participation. The official journey of Azadi Ka Amrit Mahotsav started on 12th March 2021, which starts a 75-week countdown to our 75th anniversary of independence. That is on 15th August 2022. You know, science and technology and its capabilities are fundamental for social and economic progress of a country, especially when it is developing. Therefore, Indian Council of Agricultural Research has taken up a low-cost but people-intensive campaigns on dedicated weekly theme for each of the 75 weeks. Under the ICAR umbrella, NRRI is organizing this Azadi Ka Amrit Mahotsav lecture series. Today, we are lucky to have an eminent scientist, Professor Yinong Yang, with us to deliver this important lecture. It was 2012 when the seminal paper on CRISPR-Cas9 was published by Jennifer Doudna and Emmanuel Charpentier. After a very short span of time in 2013, as one of the pioneers in plant, Professor Yang has demonstrated successful application of CRISPR-Cas9 for rice genome editing. And you know, very well covered in international media, the famous case of genome-edited non-browning mushroom is from Professor Yang's lab. We are really lucky to have you here, Professor Yinong. So now directly going to the main program. Now I would like to request Dr. P.C. Roth, Head Crop Protection Division, NRRI, to formally welcome the guest speaker and brief about the program. Roth, sir, please.
Good evening, Professor Yinong Yang and colleagues in USA, and a good morning to everybody in India. I take this opportunity to extend a warm welcome to Honorable Yinong Yang, Professor, Department of Plant Pathology and Environmental Microbiology, Huck Institute of Life Sciences, Pennsylvania State University, USA, to our institute, though in virtual mode, on the occasion of a special talk series to celebrate 75 years of India's independence. I welcome our Honorable Director, Dr. Padmini Shwai, to preside over this virtual function. Hearty welcome to all the dignitaries of ICR headquarters and other ICR institutes, head of divisions, and scientists of ICR NRRI to this virtual function. Madam, we have in this Azadi Ka Amrit Mahotsav special lecture series already organized 2 lecture series. First one is on 18th August 2021 on agripreneurship-led extension The speaker was Dr. P. Chandrasekhar, Director General, National Institute of Agriculture Extension Management, Hyderabad, India. The second lecture was organized on 7th of the September 2021. The topic was the genomics of wild and domesticated rice. The speaker was Professor Robert Henry, Professor of Innovation in Agriculture, the University of Queensland, Australia. And this is the third lecture in the series of special talks to be celebrated in the 75th year of independence, Azadi Ka Amrit Mahotsav. And the speaker is Professor Yinong Yang, Department of Plant Pathology and Environmental Microbiology, Hawk Institute of Life Science, Pennsylvania State University, USA, on topic CRISPR Cas-enabled crop precision breeding and disease diagnostic. The rice, as we know, rice is a model plant for monocots and one of the most important food crops for over half of the world's population. Rice blast, Magnaportha oryzae, and sheath blight, Rhizoctonia solani, are the two most destructive diseases that negatively affect worldwide rice production. Professor Yang's research program aims to understand the molecular mechanism of pathogen virulence and host immunity during the rice fungus interaction, and to translate basic knowledge into agricultural productivity via genome engineering and biotechnology. An anti-browning mushroom developed by Professor Yang using CRISPR-Cas gene editing technology was named a 2016 Best of What is New Award winner in the engineering category by Popular Science magazine in USA. Professor Yang kindly accepted our invitation to deliver this special talk on this topic, CRISPR-Cas-enabled crop precision breeding and disease diagnostics on virtual mode. I firmly believe this lecture will serve as a source of knowledge and strategies for the scientific community. Again, I welcome you all to enjoy this special talk by Professor Yang. Thank you.
Okay. Can you hear me, I guess? Thank you very much, Dr. Ross and Dr. Mola for the welcome remark and kind introduction.
No, so you need to wait for a few
Oh,
minutes,
sorry?
because
Okay.
we have something in between.
Oh, okay. Yeah.
Okay, so Shudhamani, please go ahead.
Yeah.
Thank you very much, sir. Although most of us know Professor Yang and his great contribution, for many participants from other institutes and students, I request Dr. Sudhamoy Mandal, sir, Principal Scientist, NRRI, to introduce Professor Yinong Yang to the audience. Sir, please.
Yeah. Ladies and gentlemen, good morning, India, and good evening, USA. Yesterday's Science magazine headline was, "Gene editing produces all male or all female litters of mice." This is what is called CRISPR phenomenon. CRISPR, clustered, regularly interspersed short palindromic repeats, is an adaptive immune system used by microbes to defend themselves against invading viruses by recording and targeting their DNA sequences. Only 9 years ago, scientists reported that CRISPR-Cas technology can enable precise and efficient genome editing in living eukaryotic cells. And we know since then, the method has taken the scientific community by storm, with thousands of labs using it for applications from biomedicine to agriculture. And 2020, the Nobel Prize in Chemistry was awarded to the pioneers in this field, Emmanuel Charpentier and Jennifer Doudna. Today, we have with us Professor Yinong Yang, one of the pioneers of genome editing in plants using CRISPR-Cas system. Professor Yang is a professor in Department of Plant Pathology and Environmental Microbiology, Huck Institutes of the Life Sciences, Pennsylvania State University, USA. He holds a BS in biology from Zhejiang University, formerly Hangzhou University, China, MS in botany from University of South Florida, USA, and a PhD in plant molecular and cellular biology from University of Florida, USA. His expertise include molecular plant microbe interactions, disease resistance and abiotic stress tolerance, functional genomics, biotechnology, genome editing and engineering, precision breeding and crop improvement, gene edited and genetically modified organisms. Professor Yang has more than 100 very impactful research papers and more than 10,000 citations with H index 38 and I-10 index 60. A recent news in 2021 news article states, featuring Professor Yang, CRISPR-Cas technology could enable early diagnosis of devastating citrus disease. Really, a Penn State-led team of scientists has developed a diagnostic test for early detection of citrus greening, a disease that causes green, misshapen, and bitter citrus fruits, making it unmarketable and threatening the $3.35 US citrus industry. And as mentioned earlier, in April 2016, Nature writes about Professor Yang's findings. Gene-edited CRISPR mushroom escapes US regulation. Professor Yang engineered the common white button Agaricus spiceporous mushroom to resist browning. The effect was achieved by targeting the family of gene that encodes polyphenol oxidase, an enzyme that causes browning. By deleting just a handful of base pairs in the mushroom's genome, Professor Yang knocked out one of 6 PPO genes, reducing the enzyme's activity by 30%. We are indeed delighted to hear Professor Yang on CRISPR-Cas phenomenon that has taken the scientific community by storm. Sutapa, please.
Thank you, sir. Now I will request Professor Yang to deliver his lecture on CRISPR-Cas-enabled crop precision breeding and disease diagnostics. Professor Yang, please.
Okay. Thank you very much, Drs. Ross, Mandel, and Mola for the welcome remark and kind introduction. So it is my great pleasure to present this talk in celebration of 75 years of India's independence. Like China, India are a huge country. Obviously, rice is also a very important crop, both in India and China and a lot of Asian countries. So today, my talk will kind of primarily focus on rice research have done in my lab related to the CRISPR. I will kind of briefly also introduce the disease diagnostics we have done with the citrus green pathogen, and a little bit about the genetically modified mushroom and relevant issues related to genetically modified organism or GMO. Before I start, it looks like, hello. Are you able to hear me?
Yeah, it's moving.
Oh, sorry, I guess I kind of, my screen kind of freeze a little bit. I couldn't advance the slides. Okay. Yeah, so before I start with that genome editing, I just want to briefly introduce the concept of genome editing for those who may not be familiar with CRISPR genome editing. So genome editing conceptually is basically a pretty simple, is involving break, repair, and modify the DNAs. So there are a lot of agents like chemical mutagens, radiations can break DNAs, but those are kind of random mutagenesis. However, in order for genome editing to occur, you need break DNA at a specific location using different kind of tools such as CRISPR and early tools, single finger nuclease and TALEN nuclease. So upon the creation of double-strand break, all the living organisms have a different DNA repair pathway that can repair the double-strand break. The most two common the DNA repair mechanism is a non-homologic end joining mechanism, which is predominant in high organisms, particularly like animals and plants. In this case, double-strand break will be simply ligated by a ligase, and during that process, a small insertion or deletion will be introduced. That results in introduction of a stop codon, a frame shift and introduction of stop codon, and effectively knock out the gene. This is a very good tool approach for loss of function analysis to knock out genes. Another common DNA repair pathway is homologic dependent repair, which is more predominant in bacteria and fungi. In this case, a donor template are needed to be introduced in vivo. And donor template will serve as a template for subsequent DNA synthesis, and that leads to that homologic dependent repair. And eventually, so either a replacement of a DNA sequence or insertion of a DNA sequence. And this process is very precise. It's a designed process. It's a designed strategy for genome editing. However, this is a very low efficient process. So one of the major goals in my lab actually is also involving how you can using either HDR or other method to precisely insert the gene or replace the nucleotides. So as the previous speaker have introduced, CRISPR is a new tool for genome editing. In nature, CRISPR-Cas actually is a bacteria defense system. So it's just like a DNA restriction system. So like restriction enzyme system is another bacteria defense system. So CRISPR stands for cluster regularly interspace short palindromic repeat, and Cas is CRISPR associated nuclease. So CRISPR itself, as shown here, this dark shadowed repetitive sequence. These are sequences not really involving use for any genome editing, although we always call it CRISPR editing, or but the CRISPR the itself is not used for genome editing. Rather, that CRISPR associated nuclease, which is a size that can make a size-specific cleavage DNA locus, and that's actually the enzyme are the ones used for genome editing. So there are a lot of CRISPR systems in the nature. I mean, virtually over half of the eubacteria and over probably 80% of the archaea all carry different kind of a CRISPR system. Generally, we can classify the CRISPR into two major classes, class 1 and class 2. Class 1 involving multi-protein components that form so-called cascade. So involving many genes actually encoding for those cascade involved in that the CRISPR cleavage. By contrast, that class 2 are usually only require a single nucleus, such as Cas9 or Cas12 or Cas13. So class 2 are more commonly used for genome editing. Obviously, the most common one is the Cas9. So in the nature, usually this is a CRISPR defense system. So you can know if the DNA phage invading the bacteria, that bacteria can actually take a small piece of the so-called spacer that insert behind that CRISPR, the leaders, and this kind of the space could be as long as like 500 spaces. So you could have this 500 repeats. So some are larger, some are small, and that array can be transcribed into a pre-CRISPR RNAs, and that CRISPR RNA, pre-CRISPR RNA can be processed by the cascade or single nucleus and to produce this CRISPR RNA, and the CRISPR RNA will able to target the guide the Cas nucleus to particular loci, especially if the same bacteriophage infect the bacteria, and then it will be degraded. So this is what happened in the nature. Then in the landmark paper, 2012 paper by Jennifer Doudna and Emmanuel Carpentier, they adapt this CRISPR system, use that for genetic engineering. So you can see in that particular paper, they basically mix the Cas protein and a CRISPR RNA and also a trace RNA. So this is a trace RNA, and so this is a two component of CRISPR. These RNAs and mix with the Cas9 and guide the Cas9 to the particular loci and result in a DNA cleavage. You can also fuse the CRISPR RNA with the trace RNA and form so-called single guide RNA, and that will also able to guide the Cas9 to a specific loci genomic site and make a double strand cleavage. So in this paper, although this originally done in vitro, but subsequently Feng Zhang's lab in MIT and many other labs have demonstrated this works in vivo in mammalian system as well as plant and microbial system. So for this invention, Emmanuel Carpentier and Jennifer Doudna received that Nobel Prize in chemistry last year. So since that 2012, the invention of CRISPR for genome editing, this tool is become broadly used and for many different kind of applications. So since that 2012, we just have one paper, and now we have about 25,000 publications related to CRISPR genome engineering, and approximately probably 10% of them are related to plant genome editing. There are many paper has been there are many company has been established, and so many companies, some established companies, also a lot of startup companies have been working on CRISPR. So some of these like work on biomedical field, such as Adidas Medicine, in Intellia Therapeutic. Both these are headquartered in Boston, and also CRISPR Therapeutic in Europe. In the plant field, we have pairwise plants in area agriculture, those in US, and also tropical bioscience, that's in UK. Obviously, there are many other companies. These are just a few examples. So those companies has been working on all kinds of the biomedical research, such as for genes therapies, drug development, animal models, and also all kinds of genetic study genetic variation. In plant field, also obviously we have a lot of lab have done genome editing for crop improvement, and also people work on like industrial microbiology and material science and biofuel, also using CRISPR to enhance their productivity. So my lab started work on CRISPR genome editing shortly after that publication of Jennifer Doudna's paper. and we published our first paper 1 year after the publication of Jennifer's Darden paper, mainly working on rice. You can see that in this picture, so you only really need two components, Cas9 and also the guide RNA in order to carry out genome editing. So the guide RNA, in this case, is single guide RNA composed of the guide RNA spacer. Usually this a 28 base pair. It's almost like a PCR primer. And also fused with the guide RNA scaffold. This originally is trace RNAs, and that's kind of part of a sequence also bonds associated with that Cas9. So this guide RNA able to direct the Cas9 to a specific loci, and based on the complementarity with that guide RNA with the spacer, with the proto spacer in the genomic site. And the Cas9 will make a cut 3 base pair upstream of the PAM. So for SP Cas9, it will cut exactly 3 base pair upstream of PAM. So that will create a double strand break. So we have used this for that plant genome plant and microbial genome editing. So for plant genome editing, particularly in rice, that's kind of basic process I can show here in this slide. So you can make a gene construct, express guide RNA and also Cas9. You can also using a so-called DNA-free IMP complex, which consists of the guide RNA and also, so could be in vitro transcribed or chemically synthesized guide RNA, and also Cas9 protein, which you can purify from E. coli. So you can just mix these two. And so both cases can be introduced in callus by gene gun, particle bombardment. If you are using binary vector, plasmid vector, you can introduce obviously using agrobacteria mediated transformation. And this usually is pretty commonly used for plant system. So after introducing the callus, you will go through selection, just like you making a typical transgenic crop. So you select hygromycin resistant callus and you regenerate plant. And then you follow by genotyping. You can examine by PCR, and you can cut with T7E1. That's the that can detect the single a few nucleotide insertion or deletion. You can also do a sequencing, Sanger sequencing. You can also do high throughput like Illumina sequencing. So after genotyping, you also do a phenotyping, whether examine some morphological change, or you evaluate some particular ergonomic traits such as disease resistance. You can also easily obtain, for rice, you can easily obtain transgene free line through the Mendelian segregation. So only you retain the edited genes locus, but you remove the vector sequence that originally integrated in the callus and the regenerative plant, and you can segregate. And those transgene-free line, you can, if those you have obtained favorable agronomic trait, you can bring to the field for field trial. So our lab have created a few tools to facilitate this process. The first one is we have done some of the analysis of guide RNA to basically predict the guide RNA in different plant species. In particular, this website, we have 8 plant species available, including rice, maize, soybean. Obviously, now there are a lot of other websites you can use, particularly the website like developed by the Huazhong Agricultural University. They have a lot of species available now. But this one we have been used early on by many labs to predict guide RNA. So you can synthesize this guide RNA based on the by selecting the specific guide RNA. So that website help you to select specific guide RNA targeting a particular locus in the plant genomes. Next step, you were able to basically ligate these specific guides into that vectors. It could be just a regular vector, or it could be binary vector that can facilitate agrobacterial mediated transformation. And the target could be a single genes, or also could be multiple genes. You can I will talk about a PTG approach we developed later on. So you can also put either single gene or multiple gene in the guide RNA into that vector, and you express those gene in the plant, and then target to either single or multiple genes. So this just shows that if you express this guide RNA in rice cell, and in this particular case is a rice protoplast. So that's kind of easy way to assay if your Cas9 is expressed. You can see here, you can detect the Cas9 by Western blot, and you know this Cas9 is expressed. And we also introduced GFP along with Cas9 in this particular case. So we know that this transformation it's pretty it's highly efficient. Then you can go on for stable transformation. If you verify this that construct works, you can proceed with stable transformation. You can see this case, we knock out a gene called MAP kinase 5, the involving abiotic stress, and you can detect by T7E1 assay that this cut are showing there are editing, and also you can subsequently you can also do a PCR to detect if there are restriction enzyme present. And if there's no restriction enzyme, you have to do T7E1. Obviously, you will follow by DNA sequence. In this case, you can see that there is a single base pair deletion, a single base pair insertion that practically resulting frame shift and knocking out these genes. So this allows for a knockout of a single gene, but in many cases, actually you require the analysis of multiple genes. So in fact, one of the major advantages of CRISPR is its flexibility to allow knocking out the multiple genes. So in order to knock out the multiple genes, you actually require a very efficient expression of multiple guide RNAs. So typically, for those who have done CRISPR genome editing, you know that guide RNA are expressed by polymerase III promoter. So like U3 or U6 promoter that will drive the guide RNA and express the guide RNA. While Cas9 usually expressed by either constitutive, usually a strong constitutive promoter, such as the ubiquitin promoter. So if you need to express multiple genes, you have to kind of stacking multiple promoters, and that quickly your construct will be very large and kind of very inefficient to transform into the agrobacteria, or even in animal system is very difficult to carry by like a viral vectors. So a few years ago, we developed this so-called polycistronic tRNA guide RNA technology for multiplex genome editing. In this case, we've taken advantage of that tRNA processing system. So this is a tRNA. We all know tRNA is a transfer RNA that delivers the amino acid for the protein synthesis. So tRNA, in order to produce a mature tRNA, the pre-tRNA has to be processed. So RNA-P and RNA-Z will cut in the 5' leader and 3' trailer to produce a mature tRNA. So we arrange tRNA and we separate basically guide RNA by tRNAs. And so we kind of alternately arrange tRNA, guide RNA, and tRNA, guide RNA. And we find that after transcription of this tRNA transcript, there can be very efficiently processed by RNA P and Z to produce individual guides. And those individual guides can target the respective genomic site. And we found there's many advantages for this. We find this cleavage is highly efficient. We hardly actually detect any intermediate. So they are pretty much completely processed. The other thing is that usually in order to highly express the guide RNA, there is some requirement for the first the nucleotide. So if you use U3 promoter, you need to start with A, so adenine. If you start with using the U6 promoter, is better to use G. But in case of using if you adding the tRNA, then there is no such a requirement. More importantly, this also allows to use of the polymerase II to express guide RNA. You know that polymerase II promoter, it's more stronger, and also you can do control the spatial temporal expression of multiple guide RNA. So you can do an inducible expression and tissue-specific expression. Another additional advantage of tRNA is that tRNA itself actually is an internal promoter, can enhance the guide RNA expression. So we find that often that the expression of guide RNA is much increased. I mean, from somewhere from 3 to 10-fold increase of the guide RNA after you adding this tRNA. So we find this, just give you an example. I mentioned this very important feature using the polymerase II. So in this work done by my former postdoc, after he returned to China in Huazhong Agricultural University, so he actually placed that the PTG of this polycystic the cassette in the intron. So he also driving this the whole exon here actually is a Cas9. So he actually have a exon encoding exon 1 and exon 2 encoding for Cas9, and put the intron the PTG in the middle of intron. And this gene are expressed by a polymerase II promoter. And in this case, the promoter can be either constitutive, inducible, or tissue specific. So he find like here, if you express with a PR gene, which is a disease passage inducible, and then it is still able to induce result in the pretty efficient cutting. And this is yeah. So this you can see this 25 to 30% of this DNA are edited using this inducible system. We also test this after introduced into rice plant. You can see this just a visual editing of the phenotype, but after editing of the phytoin desaturates. And so this case knock out this PTS gene. If you have both copy are knocked out, so it's a biallelic mutation, it will result in an albino phenotype. And we find that in our study, 87% of the editing are biallelic. I mean, there also probably some of rest of the 13% is also mostly monoallelic. So we can almost achieve a close to 100% editing of this either single and or biallelic, monoallelic or biallelic mutations. And furthermore, you could actually knock out multiple genes. In this case, we have 8 genomic loci targeted by this PTG in stable transgenic plant, and they're all edited simultaneously pretty much. And if the deletion is editing is occur on the double strand break occur exact simultaneously, you actually can't get the deletion fragment. And this is not only just applicable for knockout genes, so it can also use the same approach for other things like base editing or prime editing or transcription activation or suppression. So Dr. Coutu, when he in my lab, he actually used this system for try to do a multiplex editing using the adenol base editor. So in this case, he selects the two target, Y-strap leaf and a zebra 3 loci. So you can see here, he is trying to basically editing, change the A to G. So he put this PTG system, so tRNA alternated with that a guide RNA here and another guide RNA here, and then it combined with that adenine base editor. So you can see here he was able to get a very high efficient editing with the zebra 3. Unfortunately, the editing with the WSL5 is pretty low. So if both editing efficient are high, we can easily get the multiplex editing in both genes. Because of low efficiency here, after we screen a lot of mutant, we still couldn't get the transgenic rise line with both the gene edited. However, we got both individual editors, obviously. So you can see here, A become G and also A become G. So in this case, you can see the resulting edited line showing a phenotype. This case is zebra, zebra and the zebra phenotype. You can kind of strap here, and then this one is so-called white stripe leaf. It's pretty obviously you see a white stripe leaf phenotype. So this demonstrates a successful base editing, although we are not able to get both phenotype as in the same plants. This multiplex editing is not only applicable when we initially done in rice, it also works well in fungi. You can see here we did the work in rice blast fungi. In case of rice blast fungi, it's a haploid. We actually have to supply the donor template. Otherwise, if you make a double strand break, the fungi will die. So once you supply donor template, those donor template will repair through the HDR. You can see we're actually able to get all three in combination of different combination. In some cases, we got a single and double. We could also a few mutant actually got all three gene mutated. So in this case, we basically introduce a restriction enzyme site, and so we can check they are edited. So this work in not only plants and also worked on microbe. We also actually did a work in collaboration with people working on animal system. So human, in this particular case, we actually added human cells, and you can see we could get a pretty high frequency of deletions up to 40% of those genes. You have simultaneous both sides cut by that guide RNA, the Cas9, and you result in DNA fragments. And this actually result in some phenotype, which is showing that downregulation of wingless that signaling and cell proliferation. So the PTG, it seems to have a very broad application on not only in plant, but also in animal and microbial, and many other labs have used our system for various dicot, monocot, animals and mammalian system. We also did some work in microbe, and there are other labs also did work in yeast, like dictyostelium. So yeah, so this has been quite widely used in different living organisms. So now I kind of going to shifting gear to on that precision breeding using mainly rice as the examples. So CRISPR obviously can be used for a lot of the basic research for gene discovery and function genomics. It also can speed the breeding process and also very useful for hybrid breeding. I don't have time to provide example today, but if you read some recent literature, it can be used for haploid induction, male sterility, generation, pretty easy generation of that male sterile line for hybrid breeding, and it has been recently used for fixation of heterosis and broken down the self-incompatibility in potato. So it obviously can be also used for various trait improvement. So I will give some example of yield quality improvement, disease resistant, and obviously there are also many work has been recently been reported for nutrient use efficiency improvement and accelerate domestication of wider like wide tomato plants. So here's just one example for functional study. So this is a PTR gene, is a gene confer broad spectrum resistant to rice blast fungus. And USDA spent scientists spent almost 10 years to map and map clone this map-based cloning of these genes. However, they're not sure if this gene is indeed a gene responsible for disease resistance. They have actually collaborated with me many years ago and cloned a gene. We tested and found it was the wrong gene. So they went back and cloned this PTR again, and they sent me the sequence, and we find that sequence in the resistant background, and we made the CRISPR mutant line, and you can see those genes are knocked out due to frame shift. And we find that the resistant line control show resistance, hypersensitive response, but the mutant line, here you can see the two mutant line both show susceptibility to a different strain of rice blast fungus. So this is a very good example show use of CRISPR for loss of function analysis. And obviously this is a test in a kind of more reverse genetic study to study a single gene. Recently, my former postdoc went back, who actually also this a collaborative work with our group, and they made a construction of a library of mutant. Their other lab have done making basically entire library of that rice genome. But in this particular case, he is targeting over 1,000 rice receptor-like kinase, and he used a new method that don't require DNA sequencing, and you can able to tell which gene is targeted. So it's kind of a little more a clever approach. But anyway, so he made a construct this mutant library, and he found in the T0 line, he had a 5,000 T0 line, 96 or 90, almost close to 97% of all like have single tag, so single gene mutation. Some of them he has a double gene mutated, have two tag, and there are a few basic negative, false negative line, so there's no tag. But majority of them, 97%, he has tagged. So it's basically a very efficient way to generate a mutant library that not only allow reverse genetic screening, but also allow for forward genetic screening too. In my lab, we kind of interested in basically to apply these two to improve the crop yield and disease resistance. So we select a rice cultivar called Jupiter, which is widely grown in the southern United States. It is a high yield, early maturing and short stature with good milling quality. It has a moderate resistance to one particular races, but susceptible to most of other races in the southern US. So we try to improve further improve the yield and disease resistance. So we select targeting 2 genes, one called the ideal plant architecture one. A lot of people, breeder maybe know, this gene actually relates to heterosis, is a member of SPL gene family. It regulates the tillage outgrowth and panicle branch. So, so it actually, if the gene expressed in an optimal level, it can promote rice yield. So if the expression is too high, the yield is low. If it's too low, it's also low. But if expressed at optimal level, that could be achieved by heterosis. If you have a hybrid situation, they actually sometimes hit this that optimal level, and you actually also got the high yield. So we just try to modify the promoter region of this gene, because in the promoter gene, there is a GC-rich region, which is heavily, could be heavily methylated, that could lead to the downregulation. So we place some guide RNA, actually could result in disruption of this GC region. In some cases, we can see totally deleted. So there are other regions we selected that maybe we just kind of more or less random in some of cases. So we got 120 line, and we did some preliminary phenotyping, genotyping, and then we select 20 lines for further analysis and transgene-free progeny. So you can see from this figure that there are a lot of variation. First, we notice is the heading date, so that the maturation time. So normally this Jupiter rice, it takes about 110 days for the heading days, 110. But we notice some mutant have take a longer time, and there are many of them actually got the early heading days. Some are weaker, or 8 days and 9 days early, which is actually a desirable trait. The breeder actually, it was interesting to get some early maturing variety of the rice, so they can grow other crop after the harvesting of rice. So we also noticed that we do see some of line have increased yield. So this dashed line is that control. Obviously, majority of them actually have a low yield, but there are some of them actually show high yield. That's mainly is due to increased panicle numbers. There are very few cases, maybe their average panicle weight also increased slightly, but mostly is increased due to increase of panicle number. So this we also already have a transgene-free lines is ready for field trial. And obviously, those are actually done in the greenhouse. So sometimes this data is not necessarily reflection if you some of field studies. So that's kind of yield. We have not really done any work with the rice quality, but in collaboration with my former visiting scholar, we actually, so he went back to China, actually did some work with barley. And so this case, actually, he manipulated vitamin E levels. And so he targeted 2 genes in barley that responsible for the biosynthesis of 8 vitamin E isomers. And he found that by targeting these 2 genes, and so by mutating either single genes, or in some cases, he also got double mutant. In this case, he showed just a single mutation. You can actually shift the profile of that vitamin E. So in this case, you can see that reduction of those vitamin E isomer. In this case, those isomers are pretty much gone, but only retain these two major isomers. So this is this study demonstrates this CRISPR is an effective tool for metabolic engineering, altering of different metabolite, and maybe modify that the nutrition quality and maybe in this case, basically vitamin level and composition. So for rice, for me, actually, I kind of plant pathology. We obviously most interested in disease resistance. We actually also target two genes. Here is a BSRD1. This is gene cloned by a Chinese scientist in Sichuan University, and they show it's a broad spectrum resistant. And basically, this BSRD1 gene, if you this gene, if it's knocked out, a reduced expression of this gene will lead to the increased level of reduced level of peroxidase and increased level of H2O2. And so that's enhanced disease resistance. So we thought, well, maybe we want to also target the promoter of that regions. We don't necessarily want to completely destroy that gene. Sometimes may have a pleiotropic response. So we want to reduce expressed gene to see if we could actually increase the disease resistance. So we made a 8A edited line, also target this promoter region. A lot of them actually was deletions that result in low expression of this gene. We did some preliminary study recently. Some of line actually show slightly enhanced disease resistance, but we require to do more extensive study to screen all those lines to see which line have significantly improved disease resistance. Katu and also with my PhD student Justin, they're also interested in the more precise editing with base editor. In this case, they use the adenine base editor to basically targeting a gene called PID3. So PID3 dominant, if it's dominant the allele actually is the resistant. Okay, if it's in some of the line actually like D-glucose cultivar actually carry the resistant, but lots of our commercial cultivar actually is susceptible. Is because there is truncation in the natural variation. There is a stop codon there, resulting in this susceptible allele. So using the adenine base editor, we can actually convert the stop codon to a glutamine, basically. So you can see basically by changing T to C, which is same as A to G. So we actually able to add this Jupiter line and convert to T to C. So we already successfully obtained this edited line. We also got a transgene-free mutant. My student Justin is currently testing this for specific resistance to a particular races of the rice blast fungus. So, well, we talk about creating all kinds of mutant lines. So what about the regulations? So we all know that GMO, genetic modifier, especially those transgenic plants, there is a pretty high bar hurdle to go through the regulatory process. And also that around the country, there are a lot of public are very concerned about genetically modified organisms, particularly some of these transgenic crops. So we have done early on, actually, now it's like, 5 years ago, basically, we was initially one of the alumni who is the CEO of a mushroom company. He said, he was kind of interested in that some non-browning mushroom, if we could do some work with that. So we thought, okay, I just start doing some CRISPR, works great with the plant species. So I tested, so he suggests the mushroom, and Penn State, actually, our department is known for mushroom research. There's not many mushroom research in the United States. Right now, we only have 2 left in our department. So I collaborate with mushroom researcher, and he helped to provide some materials which knock out a particular gene called the polyphenol oxidase gene. And it has a 6 gene. We this study are more or less still a proof of concept. We knock out 1 gene and show a slight reduction of the browning. However, in order to get a really marketable non-brown mushroom, we probably require knock out the multiple genes. So at that time, I was presenting my research, rice research, and some of work on mushroom, potato in a meeting, and USDA scientist, aphis scientist, scientific advisor actually for the USDA aphis attended my meeting. Then he invited me to visit a USDA aphis facility in Beltsville in Maryland. And for some reason, they really interested in this mushroom story and encouraged me submit a letter to them. At that time, US have not set the policy for how to regulate CRISPR. So I sent a letter, and half years later, they come back and say, hey, if you have a mushroom, you just have a deletion of the DNA fragment or a few base pair, and that will not subject to the typical transgenic regulation. They also made the same decision for the DuPont's Waxicon. And in this case, a mutation of wax gene resulting a sticky starch. Basically, the corn is a sticky starch. DuPont have done some field tests, although they have not yet really commercialized this the corn yet. So so USDA just last year, they really formalized the rule, they call the USDA secure rule, that stands for sustainable, ecological, consistent, uniform, responsible, and efficient regulation, basically. So if you have a deletions or single base pair substitution, such as many examples I presented in our rice research and also mushroom, those will not be subject to the typical transgenic regulation. And also, if you insert a sequence, and if that inserted fragment can be also transferred through the typical, let's say, crossbreeding. So that practically, if you're dealing with some DNA from the white rice or some relative of rice, and then they will not be subject to the regulation too. So because of that rule and more relaxed rule, we actually have a couple of years ago, the first genome edited soybean marketed by that CAS-enabled. So CAS-enabled is a company in Minnesota. Although this is not created by CRISPR, it's created by so-called tail effect nuclease, old version of genome editors. So this is already commercial available in the United States. Then just a few months ago, well, just a couple of months ago, that in Japan, we have the world's first CRISPR-edited tomato on sale. That's starting on September 15 of this year. So this is a high gamma tomato. So what they did, they basically delete a C-terminal autoinhibitory domain of glutamate carboxylase. So basically make this enzyme activity very high, will be not auto-inhibited by the product. They result in 5-fold increase of that gamma aminobutyric acid. And this GABA is usually considered as a supplement to help to reduce the high blood pressure. So kind of help manage the hypertension. So this is already on sale now, and this is developed by scientists from Dr. Izura from University of Tsukuba, and he co-founded this Senatech seeds outside of that Tokyo. However, European Union still have very strict laws. So they have a rule, actually ruled in July 2018, and they said that, well, they will still treat the CRISPR-edited crop as same as that any other transgenic plant. So there's no much difference. So that's actually create a problem. Let's say if for like a rice research in rice production in US, lots of rice in US export to European Union, Middle East, and South America. If you have different law, that obviously creates a problem. Also, we have a lot of public concern too. In case of my mushroom, after my mushrooms story broke out, I got emails and also like a postcard, even this one from Canada. People kind of scared about frankenfood or obviously. So in fact, the mushroom company initially, the CEO encouraged me to do this genetic modified or gene edited mushroom. His company also got some phone calls and emails, and they actually never really direct sponsor me. They just had actually provide some donation to the Pennsylvania State University. So the university has some small grant based on their donation. I got those small grants from the university to carry out the work. So I actually never really received any company money, but still there is one story written by Scientific American. So they got some information mentioned about that company. They got some phone calls, emails, and their marketing people also was really interesting to push that organic mushrooms, which can reap a high premiums. So they're really not particularly interested in any gene-edited mushroom. However, recently I do contact by some other individual investors, some venture capitalists, and they're really interested in still doing some mushroom research. So maybe I will start to do some mushroom again. I don't know yet, but so that's the mushroom stories. Lastly, I guess, well, there's some good news recently. UK just recently that indicate they will actually not regulate gene edit the crop if you do any field trial. So in experimental staging, for example. So that's good news. However, they still will regulate if you want to commercialize that gene edited crop. But in our lab, we thought, well, so obviously, if we do gene edit for all the field trial, have all this kind of, and that the commercialization, we have all this, there's more challenge, there are many challenges. But CRISPR can also use for diagnostics. So that's the work we did the most recently. We tried to use the Cas12a in this case for early detection of citrus greening. For many of you maybe know that citrus greening is a really devastating citrus disease that resulting in like 80% yield loss, production loss in Florida. So also citrus greening pathogen is present in plant in very low titer and also uneven distribution. So it's very difficult to detect. So we demonstrate that actually you can couple with recombinase polymerase amplification process with the CRISPR and you can detect with various methods, whether using microplate reader, fluorescent detection, or you can using lateral flow strip. So this is very sensitive. Actually, we demonstrate it's almost it's somewhere 100 to 1,000 times more sensitive to quantitative PCR. It's also very specific. It's pretty rapid. It can be done in probably 1 or 2 hours. So you can see here, if you this is done by fluorescent microplate reader. You can detect all these infected sweet oranges, pomelos, periwinkle, alternative host, grapefruit, and also in salad. In some of cases, in this grapefruit, and those was originally was not able to detect by quantitative PCR. Now we can actually able to detect at a low level by that CAS-12 detection method. Another advantage is you can actually couple this CAS-12 detection with lateral flow assay. So it basically immunostrip, you just dip in the reaction buffer. You can actually reach also automolar sensitivity, which is 10 to minus 18. So this is it's basically 100 to 1,000-fold time more sensitive than the quantitative PCR. So you can see that you can detect all these positive samples, but not negative samples. However, our lab is still focused on trying to continue work on that, try to breed this Jupiter cultivar. I try to create a so-called, hopefully called ideal cultivar that can improve the yield and quality, hopefully in the later. Also disease resistance. So I try to combine multiple traits in this cultivar using different approach. I think this is compatible even with organic farming, because this basically you can reduce the use of pesticide and fertilizer, improve environment. Hopefully that will also lead in the long run, we can hopefully get the consumer acceptance, and we can improve the food security for people in the US and around the world. Thank you very much for this. Well, finally, I want to acknowledge a few people in my lab. Kabin Shah is a former postdoc who spearheaded research, and Bastian is a former PhD student. He now actually works for Inaria Agriculture, a startup, a genome editing company.
Thank you, sir, for the learning lecture. Now it's time to.
Shutamadi.
Yeah.
So he's not acknowledging. So please wait for.
Okay. Yeah. So that's yeah. Sorry, I probably taking it long. And also Katu, he's here. Katu actually did a superb job for base editing. He also did some work in their prediction. We just submitting a paper recently. And Matthew is a former student, and Justin is current student, and Vicky, who also helped with that, did actually the promote editing. And so we also collaborate with a number of labs working on this. And our research is mainly funded by USDA, and with some of funding also from NSF. Bastian actually supported by Monsanto's postdoc fellowship. Thank you very much for your attention. I will be happy to entertain your questions.
Thank you.
Thank you, sir. And sorry for the interruption in between. And now I would like to request for the questions. And this will be a discussion session. And I would request for the question.
Someone put their question in the Dropbox, Dr. Sutapa, Dr. Kutu.
Yes.
And some are raising
okay,
hand.
I think I should take care of it. So I would request Dr. Arup Kumar Mukherjee, please go ahead and unmute yourself and directly ask the question.
Professor Young, it's excellent, excellent information as well as the work you have done. And we are really enlightened to listen to you. Now, I have just two questions. That if you go for CRISPR-Cas9 editing, is there any chance of non-target editing?
Yeah, there is a small chance of non-off-targeting issues. However, I don't think the off-target is a major issue in plant genome editing. It is a more critical issue for any gene therapy. But based on the current study done by a number of other labs, they have shown that they actually did a genome editing and carefully select the guide. If you select specific guide, there's a chance of off-target is actually very low. They actually did a whole genome sequencing in rice and Rhabdopsis, and they find that off-targeting is basically indistinguishable from that, like some just variation between the plants. So, so I would think as long as you select guide RNA carefully with high specificity, that's probably not a major issue.
Thank you, Professor. Then another question is there. So using genome editing, if everybody goes for desirable crop or a crop with desirable traits, is there any threat for the biodiversity of that
biodiversity? You mean increase the biodiversity? Or?
No, I am telling that if we go for a designer crop with particular specific traits, then people will prefer to grow that crop only that edited one. Then regarding others which are present in the nature.
You're right. So yes, so well, that's kind of like we're doing some, you end up having a monoculture maybe, like in case of, it depends, in case of like disease resistant, actually we want to edit different individual plants. So we actually were envisioning maybe mix those seeds, so create a diversity in the rice field. Everything is also like isogenic line, but differ in different like race specificity, things like that. But if you're talking about diversity at a species level, I would guess, or maybe cultivar level, I mean, you could just improve different cultivars. There are like, I just saw a report in China, they find a new popcorn maize, a new cultivar was really superior ones. So you could still improve further, let's say increase the yield. I mean, you still preserve that diversity of that particular type of the popcorn. I guess, I mean, I don't think necessarily that technology will decide change the diversity. It's mainly how the researcher or agriculture researcher or maybe even consumer that maybe kind of direct or guide the directions.
Thank you very much.
Technology is just technology, yeah.
Thank you, sir.
Thank you, sir. Now I would request Dr. Laksha Goyal to ask her question.
Hello. Am I audible?
Yes, please.
My question is this, like in hybrid breeding, we generally focuses on self-incompatibility for production of hybrid seed. As breakdown of self-incompatibility can be done with any CRISPR method and any other mutagenic treatments. Can we create self-incompatibility using CRISPR-based genome editing in otherwise self-compatible crops?
I think it's possible. Well, obviously, it depends. So if we know, it's all depend on what kind of crop. And let's say in potato, so normally it's incompatible. You knock out the RNAs, and that actually basically leads to the self-compatibility. So you could actually knock in a genes, let's say if you knock in RNAs, also we know there are some other factors contribute, not just RNAs, but let's say I just using RNAs as an example. If you knock in that particular gene, you could also lead to the self-incompatibility. And if there is an allele there, so maybe there is an allele, you basically convert to self-compatible allele to a self-incompatible allele. Could be a single base pair change, and most likely maybe a multiple base pair changes. But in case of our race-specific, that PID3 as just a single nucleotide change. Yeah, I think it's possible. Obviously, knock-in it can be done too.
Okay, thank you, sir.
Thank you, sir. Now I will request Dr. Deepak Gayan to introduce him and then ask the question with a short introduction, please.
Thank you, sir. Thank you, Professor, again for a wonderful lecture. Presently, I am working on Chickpea. Is there any specific website for designing guide RNA from Chickpea?
So I'm not familiar with Chickpea, but Chickpea become, I mean, it's important crop. So there's a possibility. Maybe you should go to Huazhong Agricultural University. They have probably largest collection of the species. And also maybe there is also South China Agricultural University, there's a website. But those are the website, they maybe already have a particular genome set up. But in order for guide RNA prediction, you really don't need you don't necessarily need have their website. You could actually basically select the guide yourself. Let's say you can take a gene. If you want to target a gene, let's say a gene is a 1,000 base pair. You can take the 1,000 base pair and put it in any kinds of the online program, ask that they predict the specific guide RNA. So they will generate, let's say, a dozen or 50 or even 100 guide RNAs, and they will actually tell you which one is more specific, which one not specific. Then next step you need to do is basically you actually have to blast your the selected, let's say you select 2 or 3 guide RNA. You can blast the 2 or 3 against the genome of Chickpea, then to see if they're basically homologous sequence. If there's highly homologous sequence, that highly homologous sequence could be off target. So that's the way, that's another way you can actually also to basically assess your specificity. If you don't have anyone already analyze the guide RNA for entire genome of Chickpea. So that's one of alternative. But there might be already Chickpea genome has been analyzed.
Okay, thank you, sir.
It seems our participants are have a very keen interest on the topic and questions. Now we will I will request Dr. Nanda C to ask this question, please.
Good morning, sir. I'm Dr. Nanda. My question is, many people think that CRISPR can be applied to any crop and it can solve any problem like that. But is it that prerequisite for genome editing using CRISPR is we should have a gene sequences, non-gene sequences, and then we can edit or something like that?
Not necessary. Obviously, if you have a whole genome sequence, it helps. But in most cases, you probably can do genome editing without a genome sequence. Let's say you just PCR a fragment, let's say 1 or 2 kb genes. You just sequence that 1 or 2 kb gene, and you can design guide RNA based on that 1 or 2 base pair. And so you can design many guides. Obviously, you will not use all the guides. You probably will select 2 or 3 guides and design guide RNA. So, I mean, if you don't have whole genome, you have a slight risk you maybe end up off targeting because you don't know other homologic sequence in the genome, because you cannot do blast against the genome. You don't have the genome sequence, but you still can do CRISPR. The chance are you probably 90, I would say probably 95% that you will got specific editing even without the off target. So no, you don't need a genome sequence in order to do CRISPR.
Thank you, sir.
Thank you, sir.
I think, Sutapa, we have a question from NRRI. Dr. Sangamitra Samantra has asked about our rice portion. Can you just read that out?
Sir.
Chat box.
Yes, sir. Sir, I'll request Madam if she can ask the question.
Probably.
Good morning, ma'am. Sir, this is Parameshwaran. Am I audible?
Yes.
Ma'am,
Please
thank
go
you. Professor, thanks a lot for your nice presentation. Actually, we would like to thank for two reasons. One is your excellent presentation, and next is right now we are conducting a training session for students, especially postgraduate students, where double haploids, transgenics, and CRISPR is the major objective of the training session. And most of the students, around 20 students, they have so far listened to your CRISPR-Cas lecture, and we are highly thankful for this. Thank you, sir. So my one,
you're welcome.
Yeah, thank you, thanks a lot. So my one technical question is, we are working on microRNA 156 binding site in IPA1 gene. So we have edited 3 different indica varieties, and we also got the desired phenotypes like increase in number of spikelets, plant height, there is some change, heading date. Also, there is a few change in 5-10 days difference. So, but what the problem we are facing is in T2 also, it is not, we are not getting homozygous edits. We are getting only heterozygous or parental allele. So we are really stuck in for the past 3-4 months, we are stuck with this issue, and we don't know how to solve this. So can you just recommend or suggest few things to handle this issue?
So if you have a heterozygous, if you selfing and you look for next generation, you maybe can get the homozygous lines. So let's say if you have a single, so you have, I know you're editing the microRNA, the site. And so let's say you have edited just a single site had edited, another site is still wild type. So I assume you can selfing, and in the next generation, you should have, reassortment. You have reassortment and segregation. So you will have a certain percentage. Depending on, let's say, if you just a single site, then you will be a quarter of them. You should have double edited.
Yeah. Okay. Thank you. Thank you.
To get homozygous lines, right?
Yeah.
So
yes,
I don't
sir.
know if you have advanced to next generation or not. If yeah, you should advance to next generation and screen for homozygous lines.
Yes, sir. So we are actually advancing it into next generation. So in this case, how many plants we have to really analyze, whether 100 T2 lines or 50 T2 lines? So what actually, how to decide number of plants for analysis?
I would say you don't need many. If it's 25%, then you probably only need 20 lines. You should get a few homozygous.
Yes. Okay, sir. Thank you.
Obviously, depending on if you added the 2 side, that's kind of different story. If you added the 2 side, then obviously like 1/16. But if you're single, it should be 1/4.
So thank you. So next query is this IPA1 has a epigenetic regulation also, and the promoter has a big epigenetic load. So whether you are targeting the epigenetic profile of IPA1 promoters.
So we did a targeting, we did a place guide RNA heavily on top of the GC rich region. So we hopefully can disrupt that region, maybe increase expression. But our approach are more or less still random. We kind of look for phenotype first, then we're going to go back to see the genotype, looking for gene expression. So our bottom line is to see if we can achieve that optimal expression that is also associated with a high yield. So, I mean, obviously those material may be interesting used for further analysis to see how that gene expression is upregulated is due to the, let's say, removal of that heavily methylated region or other reasons.
Yeah. So thank you once again. I thank on behalf of my students, trainees, my team, sir, and with Dr. Sangamitra, madam. Thanks a lot for a nice presentation. Thank you.
You're welcome.
Thank you, sir. Now I'll request Dr. Mridul Chakravarti from NRRI to ask his question.
Unmute.
Sir, thank you very much for a nice lecture. Sir, I have just a query. That's now till now, genome editing tools are to utilize to just to make targeted change in the gene which is known, and also to sometime to study the gene function in this. But is there a possibility in near future to use the genome editing tool to do a allele mining kind of thing? Like in the different sequences of the genes, if they are edited thoroughly, especially the very important genes which has very important biological functions. Maybe we are till now we know the alleles which exist in nature. From that only we generally try to study the variability. But can genome editing tools can be utilized in future to create a lot of large scale variation in the targeted genes and find out even the best allele that does not exist in the nature till now? We do not know about it. So is there a futuristic scope for doing this kind of research using genome editing tools?
So you're talking about, I just make clear, so you ask some allele specific variation, right? Right. Correct. Are you asking for allele specific variation, creation of allele specific?
Yes,
not
sir.
like
So the question is that there are some genes which are very important biological functions. And for those still we study the alleles which exist in nature among the germplasm, and from that the best allele we select. But with the genome editing tool, for those genes, if we edit each and every basis, and even there is a chance that we can get a very even better allele which we still know about that exist in the nature. So does genome editing provide such kind of scope in future?
Well, so usually genome editing we require, I mean, if it's target editing, you will require prior knowledge of like what alleles and what genes and what the single nucleotide polymorphism or some of insertion deletions you have. So we make editing based on that. Obviously, genome editing can also randomly create some of insertion deletions, or even base editing, and also people can using base editing to do some like sort of evolution gene, basically change a lot of nucleotide along that gene sequences. So that will create some variations, obviously. So I guess I don't know I answer your question exactly, but genome editing definitely can help to create, I mean, even allele specific mutations also creates all kinds of variation if you want, I mean, if we already know, and also that's also if you can also create some of the more random mutations too. Yeah, I'm not sure I answer your question accurately.
Subwat gene. Until now, we only know only one of the mutations in it, which is very functional and gives the tolerance. But still, there is hope for surveying that gene throughout the gene, because those kind of genes which have a very robust effect, those kind of genes can be edited with every basis, and then there are new functions. Even we can get a better allele than that exists in the nature. That is my, even this is a kind of.
Yeah, that's possible. In theory, you could scan. I mean, you can also do a. I mean, those obviously is very challenging. It's not easy. But you can actually making a lot of mutations along a gene. You change a sequence, so that can basically replace many base pair along the sequences. But that's I guess more future type of. It's a little bit challenging right now. But if you know a particular mutation, you already know you want to change, let's say, A to G or C to T, that can be easily done with base editing. If it's a multiple sequence, then you will have to probably using prime editing. Right now, it's still not very high efficient, but you can change multiple base pair changes with prime editing.
Thank you, sir. So then I will move on to the next question by Dr. Sudhamay Mandal from National Rice Research Institute. Kindly ask your question, sir.
Yeah, it's really mesmerizing, Professor Yang, to listen to you. I have a slightly different question that basically last week, one paper has been published in PNAS, okay, that claims that it has developed a much better, powerful tool, genome editing tool. It basically widens the genome editing toolkit, that is CAS, I mean CRISPR associated transposons. I'm curious to know your opinion on this, please.
Yeah, I may not have seen that particular paper. Is this a paper on plant system or is it actually in the animal?
Animal system. It's animal system.
Is it transposase? You say?
Yeah, exactly.
Yeah. Well, there are a few paper published. Obviously, Feng Zhang also published transposase paper in Science a few months ago. So basically, so there's two kind of area developed. One is a lot of people is looking for a new type of Cas proteins. So some are very small, compact ones. Some people even, Feng Zhang also another paper, they demonstrate can go back to the progenitor of Cas9. Basically, those the bacteriophage, the really ancient progenitor. They demonstrate some of this Cas also works. So that's kind of develop a new type of Cas protein. Another, in terms of you talking about the transposase, is basically people try to fuse a functional domain with Cas, whether it's a transposase, also older one for receptor, like for the activation repression. Transposase is mainly people try to use that for knock-in. It seems like initially work demonstrated in bacteriosome, it works very well. Biofuse Cas9 with the transposase can significantly increase the site-specific insertion with bacteria. But then recently, there are paper by Feng Zhang, and maybe also this paper, PNAS paper, which I have not seen, demonstrate can also improve the knock-in in mammalian cell. The transposase fusion with transposase has not yet been, I guess, has not been demonstrated in plant yet. Maybe there are some lab already working on it. Our lab was initially interested in working on a particular type of the transposase, but we never really get that started. But yes, I think it should work. I mean, I'm kind of positive with some, obviously, the work already published in Science and PNAS for the mammalian system. Whether it works in plant system, I don't know exactly. I think it probably also works too, but maybe need some tweak, need some modification in order to work well in plant.
Thank you, sir. Sir, if you are, with your permission, if we can go for a few other questions. As the participants are very interested to know more on the topic, shall I take some more questions?
Yeah, I would be happy to answer any questions. I mean, I
Thank
can
you,
stay a
sir.
little longer. That's no problem.
Thank you, sir. I'll request Debashmita Panda to ask her question, please.
Hi. Thank you. Thank you, sir. And my question is, how do you screen the T0 plants?
Screen edited plan, or could you repeat the question? Sorry.
Is how do you screen the T0 plants?
Oh, T0 plan. There are many ways to screen. So it depends on how initially you design the guide RNA. Sometimes we want to kind of little easy to screen, or we design the guide RNA, make it the cutting site, let's say 3 base pair upstream of pan. We actually put in the actually design along the restriction site. Then it's very easy to, you can screen, you just extract total DNA and you amplify PCR, you make a restriction digest to see if it will cut by the restriction enzyme or not. If it does not cut, it's edited. If cut, I mean, it depends on, yeah, if it's cut, it means that is not edited. But another common approach, obviously, you do T7E1 screen. You have to purchase that enzyme, and that basically it's detect the hetero duplex. But if you have a good findings, maybe it might be easy just to you can adding adapter, and you maybe can do Illumina sequencing, some amplicon, some type of amplicon sequencing, and you can mix a lot of sample together. You can, let's say, can mix actually hundreds of different samples together by distinguished by the tag. You have PCR, you have different slide, different tag, then you can sequence them and analyze them. So that's also a very efficient ways. Yeah, for the T0 screening.
Yes, sir. Thank you. Thank you, sir.
So next,
there's one publication in by a group in China. I think they call the High Tom. I can find later on if you interested in that paper. That was for amplicon sequencing, how you can efficiently characterize if you have deal with a larger number of the edited plant. You can do amplicon sequencing and mix them and barcode them basically.
So the next question is from Dr. S.K. Das from National Rice Research Institute, but I think he was
Suttappa, you are not audible. Sushant, you can directly ask.
Sorry, actually, I was having some issues. Actually, thank you, Professor, for your fascinating lecture. Actually, I have some questions related to the editing of IPA EPA1 gene, where you have shown that the panicle size has increased. Now, my question is, whenever you are editing IPA1 gene, that the panicle size increases for the further the telar number or the panicle number that is decreasing, because there are some reports of pleiotropic effect of IPA1 gene for reduction of telars. This is my first question.
Yeah, so obviously that IPA1 regulate the telar, the outgrowth. So the telar number is so it depends on the expression, high expression, low expression of the IPA1. It will have more tiller or less tiller. So I think you actually don't want to have too many tiller. You also don't want too few tiller. So that's why it will have to have an optimal expression of the IPA1 in order to achieve the balance. So if there are too many tiller, then I mean maybe those tiller have each one of them maybe have a little lower weight and the panicle weight. If you have too few, then just have too few. The total yield is still low. So it has to be balanced.
Thank you, sir. My second question is that if you are just editing some particular gene, then that might be impact on other physiological traits.
That's possible, yeah.
Yeah. Is there possibility to just have multiple editing so that this effect could be neutralized?
Well, unless you know how that effect can be neutralized, then it will be trial and error. I mean, so you don't know. But yes, especially if you knock out the gene. So in this case, IPA, actually we did not knock out gene. We basically can turn up or turn down the expression. So it probably have a less, I would say, the pleiotropic effect. And also for other genes, if you knock out the gene, yes, you maybe have a more drastic pleiotropic effect, which is not desirable. So in a lot of cases, you maybe only want to change a few nucleotides. I mean, some just let's say, basically did not really knock out the gene, is basically some SNP changes, which maybe change a few new amino acid. Let's say from proline to threonine, or yeah, just a few change of amino acid, and that actually could affect the protein. There's one recently published paper. They demonstrate if you knock out the gene, yes, it comes for disease resistance, but it actually the plant growth was impacted. However, if you just change the one amino acid sequence, it still enhances disease resistance, but without the yield penalty. Okay, so yeah, there is a possible. Well, that requires a lot of basic research how that gene functions and what kind of mutation. So you could maybe study the lot of rice line and maybe screening and find out if a particular polymorphism contribute to a particular trait without let's say yield penalty in case of disease resistance. So I think it's still possible, but they require a lot of basic research. Knocking out the gene maybe not the best approach in many cases. You maybe just want to change the sequences slightly rather than just knocking out the whole gene.
Thank you, sir. Next question is from Dr. Samant Rai from National Rice Research Institute. So due to lack of time, I would like to read out the question. Her question is regarding, can we employ double haploids approach for getting homozygous edits or lines, since National Rice Research Institute has developed robust protocol for double haploid development through androgenesis?
Yeah, definitely. So if you have if you already create a double haploid through, let's say, pollen culture, and these are homozygous, and you don't have a problem. Let's say if you, most time you don't have problem, even with the heterozygous line, you probably still, a lot of them allele are same, but if it's double haploid, they are kind of identical. So if you design guide RNA, it will, I mean, usually will cut both. If you can express guide RNA efficiently, it was more likely chance you will basically edit. Or if it's base editing, let's say, it will more likely, mean, you got, yeah, yeah, double haploid is good material, still a good material for genome editing for sure.
Thank you, sir. The next question I'll take from Dr. Lambodar Bera from National Rice Research Institute. That is, how many genes of same trait of different traits can be edited efficiently at one time?
Well, we edited like 810. I think there's one report. I'm talking about knocking out, which is relatively easy. Basic target mutation. Yeah. I see their report. I think people did 24 genes. Let's see. Edited simultaneously, 24 genes. So I would say probably, yeah, 20 genes probably doable. I'm not sure that 24. Yeah, I mean, it depends on what type of editing. If it's just knockout, I would say probably 20 is possible. And also it depends on how, if these are redundant gene, let's say sometime if you have a gene family and you could have one guide RNA target 10 genes simultaneously because they're all same sequences. But I'm talking about unique sequence. If you have a unique sequence, you have to design unique guide RNA. I would say probably 20 is probably approaching limit. Or there might be a new method invented to allow more gene targeted simultaneously. We did about 8 or 10 simultaneously.
So will there be, sir, then thank you, sir. In continuation with the same question that has been asked by Dr. Dayal Das, is there any adverse or off-target effects on editing of multiple genes?
Yeah, definitely. If there's more gene, more guide RNA introduced, you will have a increased chance of off-target for sure. Yeah, you will increase the frequency of off-targeting if with the increased number of guide RNA. And also you have a more chance if that gene, you edit a different plan, you have a different kind of the outcome, maybe have translocations, fragment deletions, you have more kind of these unpredictable outcomes. I mean, some may be predictable.
Thank you, sir. Another question is from Subhashish. What about choosing POL2 promoter instead of using OSU3 promoter to express the PTG system in rice? That is what his question is.
POL3 can be used. POL2, maybe one thing, it could be stronger promoter. I think more important if you want to do, let's say, in some cases, tissue-specific inducible expression. In most cases, you probably not useful for any breeding purpose, but for some of other research purposes, there might be an interesting, especially in animal system or some of the model system, or some basic plant biology research. You may be interested in do some inducible editing or inducible activation or repression. Most time, if you want to do a deletion insertion, I think most people don't care that much if inducible or tissue specific. For breeding, it probably is not that much useful, but for probably more basic research type of things, inducible and tissue specificity probably is an advantage associated with the use of the POL2 promoter.
With this, we come to the end of our question and discussion session. As due to with this, thank you, sir. With this, we come to the end of our discussion sessions. There are many questions coming up, but sorry, because of the shortage of time, we'll not be able to take them up now. And we will proceed to our next. We will proceed further. That is, I'll like to request our director, Dr. Padmini Swain, for her presidential address. Madam, please.
First of all, let me congratulate Professor Yang for a very excellent, inspiring and educative lecture on CRISPR-Cas technology. And in very good morning, bright good morning in India, of course, midnight in USA. And I could see that your lecture have attracted more than 100 participants, including our researchers, scientists, students, and many more dignitaries in the country to participate. And I thank all of them for their interest to participate in your lecture. I also congratulate our team of scientists who organized this excellent lecture today morning. Professor Yang, you have delivered a very wonderful talk covering many areas of CRISPR-Cas technology, assembled gRNA Cas9 construct, polycystonic tRNA, gRNA, PTG technology for multiplex genome editing, and that adenine base editing, AT2GC in rice plant, PTG-enabled multiplex editing in rice blast fungus, application of PTG in plants, animals, and microbes. Your talk was excellent on CRISPR-Cas-enabled precision breeding, gene discovery and functional genomics, speed breeding, yield quality improvement, many more applications. Of course, you have also covered some of the other crops like talent-edited soybean, CRISPR, guava tomato, vitamin E level manipulation in barley. So you have delivered really excellent talk. And the most interesting was editing rice cultivar Jupiter, mostly available in southern US, IPA promoter edited that Jupiter rice line for from this total grain weight, panicle number, variation in all these things, and also for disease resistance. So you have delivered an excellent lecture. I profusely thank you, Professor Yang, for staying up midnight to early morning in your place to deliver this wonderful talk. We can't not thank you enough. I believe our scientists, students, and researchers had a great time listening from you about genome editing and CRISPR-enabled disease diagnosis. The world has already started realizing the tremendous potential of this CRISPR-Cas technology in basic biology, healthcare, and agriculture. Few days ago, I was surprised to see a new article that Victoria Gray, a patient with sickle cell anemia, is being treated with CRISPR medicine. So it is really amazing. I understand CRISPR-Cas has a lot to offer for agriculture and food. For realizing the full potential of genome editing technology, social acceptance are crucial. In addition, ease of regulations of crops generated through these new breeding tools are vital. DBT India has formulated a draft of regulations guidelines for genome edited crops. We hope Government of India accepts DBT proposal to exempt genome edited crops that doesn't have foreign DNA from the existing genetic engineering regulations. I highly appreciate Professor Yang for his precious time. Thank you very much, Professor Yang. I also thank all the participants and for their queries they could clarify from your talk. I really thank all of you. Thank you.
It's my pleasure. Thank you very much.
Thank you very much.
Thank you. Thank you, madam. Now it's the time for the formal vote of thanks. For this, I would like to thank Professor Yinong Yang to accept our invitation to share his experiences in the journey of learning this new breakthrough technology and applying it for the advancement of science. I, on behalf of my institute, express my sincere gratitude towards Professor Yang to spare from his valuable time for the lecture in the celebration of the Azadi Ka Amrit Mahotsav, that is commemorating 75 years of independence of India. It's about 1 a.m. now in USA and chilling outside. Special thanks for your patience for the long question round session and passion for spreading the light of knowledge and make us learning the tits and bits of the technology. Thank you, sir. Thereafter, I would like to thank our director, Dr. Padmini Swine, for the continuous encouragement for making this event happen successfully. I would then like to thank all the heads of the divisions for their support and keen interest to make it a success. I thank all the directors and dignitaries from other institutes of ICR, Professor K.C. Bansal, former director NBPGR, scientists from NRRI, National Rice Research Institute, and other institutes of ICAR, which I could see participated and joined us in large numbers, which crossed more than 140, reveals their keen interest on the topic and passion for learning. Thank you all for joining in such a large number and making this event a grand success. Now, I take the opportunity to thank Dr. P.C. Rath, Head Crop Protection Division, National Rice Research Institute, for his continuous support for arranging this lecture series for the commemoration of 75 years of India's independence. I express my heartfelt gratitude to Dr. Sudhamai Mandal, Principal Scientist, Crop Protection Division, and Dr. Kutub, Scientist, Crop Improvement Division, for their sincere cooperation and coordination for arranging such a learned lecture for all of us. Special congratulations and thanks to all our students who connected with us for the purpose of this learning. Special thanks to Head of the Division, Social Science Division, for creating a platform where more than 140 participants could join for the purpose of this great learning. Thank you one and all, and have a nice day. Good night, sir.
Thank you.
Thank you.
Thank you.
Good night, Yinong. Good night.
Thank you.